Hi, my name is Scott Evans, and I am the Director of Basic and Translational Research at MD Anderson, the Department of Pulmonary Medicine. Actually, will be assuming the chair of that department in a couple of weeks. And my lab is focused on pneumonia. Pneumonia is a worldwide threat. It results in greater quality of life years lost to disability than any other condition. That's true in every population. However, interestingly, most people don't die of pneumonia, despite the fact that we aspirate and inhale pathogens every day. So what my lab does is focus on trying to understand how the host cells sense and respond to pathogens they encounter and prevent us from getting pneumonia. We have a wide range of studies in the lab, and they range from such things as trying to interrogate those host responses diagnostically to trying to understand and mitigate immunopathology associated with robust responses to pathogen. But most of what we do focuses on trying to therapeutically induce those native responses that should be protecting us against pneumonia in a way that helps boost that protection to mitigate the risk. We have shown the remarkable capacity of lung cells to sense and respond to pathogens and generate antimicrobial responses that protect against pneumonia. And most notably, we have identified a combination of synthetic innate immune ligands which can protect against everything we've ever tested. Every pathogen we've ever trialed, from gram positives to gram negatives, fungi, viruses, nosocomial bugs, bioterror bugs, everything we've ever tried, we can protect against. And this is a really remarkable phenomenon that we call inducible epithelial resistance. And in this phenomenon, if we give these ligands individually, they really don't do much of anything at all. But given together, we generate almost complete protection, again, against almost everything we've ever tried. And even more amazingly, we find that this protection is generated by lung epithelial cells, as you see in the diagram here, as opposed to the leukocytes that we typically associate with antimicrobial responses. And that's really good news for patients like the ones that we see at MD Anderson that often have severely impaired leukocyte-mediated immune systems. We are translating this phenomenon for clinical use. We now have FDA approved trials of this agent, of this strategy, in a number of different populations. That includes trying to prevent infectious exacerbations of COPD, trying to prevent progression of viral pneumonias in patients with leukemia and stem cell transplants, and most notably at the current time, we have two different trials approved in patients with COVID exposures, either newly diagnosed patients with COVID or close contacts of people with COVID. Those trials are actively enrolling right now. I can't share any of the data yet, but we are very excited about the translational capacity of this intervention. While the translational work is ongoing, my lab is largely focused on understanding how this protection is achieved and in trying to understand how this synergy occurs. On first hearing, I imagine to many people this sounds principally like immunology or microbiology. And certainly we have trainees in the lab that do those sorts of things. But we also have BCB students in the lab, because so much of what we do is trying to interrogate the cell biology of the lung epithelial cell, and also trying to understand the complex biochemistry involved in the signaling events that we study and in the antimicrobial responses, and specifically in the microbicidal responses that we generate. As outlined on this slide, we have quite a broad range of studies that our students participate in, and open projects for students that are seeking a lab now. As I alluded earlier, there are studies on things such as diagnostic interrogation of the host response, but I think principal interest to the BCB student would be the work that we're doing on sensing and responding to pathogens and characterization of these antimicrobial responses. We have, through a number of advanced biochemical strategies, identified at least 2 different novel pattern recognition receptors that we are yet to publish, but we're very confident in these outcomes to help explain this striking synergy that is not explained by the current literature.
30 seconds.
The synergistic response that we observe is a form of coincidence detection that requires advanced biochemistry and cell biology understanding. And we spend a lot of our time trying to understand how that signal is propagated. But as I mentioned a moment ago, we also are looking to better understand the biochemistry of how these microbicidal responses are generated. And that involves things like redox biology, understanding of electron transport chain biology, and better understanding how epithelium-generated antimicrobial peptides interact with the pathogens. So I would be delighted to answer any questions because I'm excited to hear what students are wondering from the BCB group. And with that, I thank you for your attention and I'm pleased to entertain questions.